Hello and good morning, and welcome to Military Medicine, COVID-19, and the Changing Scope of Operations Symposium, the first of 4 symposia CIMVR will be hosting throughout the month of October. Before we begin, let's acknowledge the Indigenous peoples of the lands that we are on today. While we're meeting virtually, I'd like to take a moment to acknowledge the importance of the land, which we each call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and to improving our own understanding of local Indigenous peoples and their cultures. Before I introduce our symposium moderator, I will invite Dr. Stephanie Belanger to say a few words.
Bonjour et bienvenue à ce symposium sur la médecine militaire, la COVID-19 et l'évolution et la portée des opérations. Je suis Stéphanie Bélanger, je suis professeure au Collège militaire royal du Canada, fondatrice et directrice associée de l'Institut canadien de recherche sur la santé des militaires et des vétérans. Nous sommes honorés d'organiser ce symposium et nous souhaitons que vous allez pouvoir y vivre une expérience enrichissante qui va vous inspirer dans vos recherches ultérieures. Bon symposium.
It is now my esteemed pleasure to introduce Lieutenant Colonel Andrew Beckett, Trauma Advisor to the Surgeon General, Chief of General Surgery for the Canadian Armed Forces, Assistant Professor at the University of Toronto, Assistant Professor at McGill University, and Medical Director of the Trauma Program at St. Michael's Hospital, who will be our moderator for the morning panel. Lieutenant Colonel Beckett, please.
Well, good morning, everybody. It's a great pleasure to be here and be your moderator for this very impressive symposium. We've gathered some of the best researchers in the country that are doing work with COVID-19 to speak today, and so we're very excited to bring all these speakers to you, and in particular into this forum where there's many people from the military community, and we've gathered some speakers that do research in the area that directly impacts the Canadian Forces. So it's my great pleasure to bring them all today to you, and we'll be taking questions at the end of each speaker, and I'll be moderating your questions. So again, we're looking forward to some great talks today, and it's very exciting to have you all with us today.
Ladies and gentlemen, I hope everyone is well and healthy. My name is Homer Tien. I'm a former member of the Regular Forces, a current Reserve Force member, a trauma surgeon with Sunnybrook Health Sciences Centre in the University of Toronto, and I'm the CEO of Orange. Orange is Ontario's air ambulance and critical care transport provider. Most importantly, however, I'm a CIMVR fellow, and this is why I'm here today. I'm going to speak about operational issues facing the Canadian Armed Forces and how rapidly they can change. The fact that my kids are only starting their second week of school after the longest March break in history, and the fact that this is a virtual symposium, speaks to the rapidly changing nature of military operations in 2020 and life in 2020 in general. As a CIMVR fellow, my job is to encourage and engage like-minded researchers to develop research networks to help counter the new health threats facing our military personnel and their families. Over the span of my career, I've seen these threats change. They're as diverse as the missions that our troops are deployed on. The good news is that there's a tremendous overlap in the operational health issues facing EMS organizations, hospitals, and defense organizations, both nationally and internationally. EMS organizations face some of the same environmental challenges that our Canadian Forces Health Services personnel face on deployment. They need to care for patients in harsh environments inland, cold weather, hot weather, mountainous terrain, forested areas. They also need to be able to respond and treat patients on land, in the air, and on water. Hospitals face the same issues as military combat support hospitals like the Role 3 Multinational Medical Unit. Medical care is medical care generally, and ICU care is ICU care generally. We need to work together to answer these questions, and CIMVR is a natural forum for this collaboration. As a regular force combat surgeon at the Role 3 Hospital in Kandahar, I face the same clinical problem as I do every day as a trauma surgeon at Sunnybrook Health Sciences Center. How do we resuscitate massively bleeding trauma patients? I remember when Prime Minister Harper first announced that Canada would deploy to southern Afghanistan starting in January 2006. As a result, from 2006 to 2011, warfighting became the major operational issue facing the Canadian Armed Forces. And as such, massive hemorrhage became one of the major health threats facing our soldiers. And so the resuscitation of bleeding patients rapidly became the focus of our attention. In response, we built pre-hospital research networks. Canadian military researchers work with civilian academics, DRDC scientists, and our allies in the US, UK, and Israel. We looked at whether or not blood could be safely transported far forward with our special operations forces and transfused by them in the pre-hospital setting. Lieutenant Colonel Beckett and Dr. Bastarino at DRDC looked at extreme scenarios. Would blood products even survive the hypobaric conditions of a high altitude, low opening parachute jump from 30,000 feet? And once the blood landed, would it survive another 2-hour trek on someone's back, in someone's backpack in hot weather? We also studied tactical combat casualty care techniques, including tourniquet application, and how to best look after patients on the battlefield while being shot at. I have no doubt that this research helped save the lives of many Canadian and Allied soldiers, but it also informed the practice of many Canadian EMS organizations. Our issues overlap. Many EMS organizations like Orange learned from this military experience, and now tourniquet use is used to help stop extremity bleedings for most EMS organizations, and we also carry blood products to transfuse far forward. We also built research networks consisting of hospital-based researchers, military researchers, and DRDC scientists. The Surgeon General's research program funded Sunnybrook Health Sciences Center to conduct the world's first RCT looking at one-to-one-to-one transfusion strategy for massively bleeding trauma patients. Then, using lessons learned from this pilot trial, both the Canadian Surgeon General and DRDC contributed another $1 million to the PROPER trial, a US-Canadian multi-centered randomized controlled trial that made one-to-one-to-one the standard of care worldwide. As soldiers will always face the prospect of combat operations, Canadian military researchers, led by the likes of Lieutenant Colonel Beckett, still continue to build research networks with organizations like Canadian Blood Services and academic health sciences centers to develop better resuscitation and transfusion strategies, like using fibrinogen concentrate and freeze-dried plasma. Shortly after the last Canadian soldier left Afghanistan, the West Africa Ebola epidemic began. This epidemic began in 2013 and ravaged Guinea, Liberia, and Sierra Leone. A public health emergency of international concern was declared in August of 2014, and in response, Canada launched Op Serona. 79 Canadian Forces Health Services personnel deployed to a UK-led health facility in Carytown, Sierra Leone. Canadian Forces Health Services personnel worked alongside their UK military counterparts to treat Ebola patients. They had to learn how to work safely in their PPE despite the hot climate and despite the significant risk to their own safety from treating these highly contagious patients. Although we thankfully never saw an Ebola case here in Canada, we all had to figure out how to work in an Ebola environment. At Orange, like our military counterparts, we learned how to work in our PPE and learned that it isn't easy transporting patients in full PPE. My Orange paramedics faced the same problems as many of our military medical technicians and healthcare providers during this epidemic. How long can they safely work in PPE when it's 30 degrees Celsius? How can we safely transport these highly contagious patients by sea, land, or by air, wearing PPE in these challenging environmental conditions? Another research network emerged to develop best practices for caring for highly contagious patients, like Ebola patients in hot weather conditions. This team is led by Dr. Rob Fowler, who's an internationally renowned clinician and scientist at Sunnybrook, and Dr. Len Goodman at DRDC Toronto. Rob was first on the ground in West Africa as a member of the World Health Organization in 2013. With Dr. Goodman, Dr. Fowler was able to take advantage of the unique environmental facilities at DRDC Toronto, the heat chamber. The team's work is ongoing, but will hopefully yield best practices for safely and effectively caring for highly contagious patients in hot weather conditions. Of course, our new reality in 2020 is COVID-19. This is not only the major operational issue facing the Canadian Armed Forces, but for the entire country and the entire world. Thousands of CAF members deployed to long-term care homes in Ontario and Quebec. Like everyone everywhere, they wondered what was appropriate PPE for this scenario. N95 masks, face shields? Do they need to be worn only for aerosolizing procedures? Do they only need to be worn for COVID-19 positive patients? And what about aid to the civil power scenarios? What if a remote community was hit with COVID and a large number of patients needed evacuation by the Royal Canadian Air Force? How does one safely transport COVID patients by air? At Orange, we wondered the same questions. We developed and published on our PPE strategy. We also reviewed the literature on air transport of COVID patients. There are several modes of oxygenation and non-invasive ventilation that seem beneficial for treating COVID patients. They might prevent the need for mechanical ventilation and intubation, but they're potentially unsafe in the transport environment. These are high-flow oxygen therapy by nasal cannula and BiPAP. A group of Sunnybrook and Orange researchers, led by Dr. Mike Peddle from Orange and Mr. Hamid Avari, an engineer at Sunnybrook, are conducting a study on dispersion of aerosolized particles during air transport. We aerosolize saline into 1-micron particles using a simulator during flight in our fixed-wing aircraft. We measured the dispersion of the particles throughout the aircraft using a particle counter. We compared the dispersion using different modes of treatment, including mechanical ventilation, high-flow oxygen, BiPAP, and nasal prongs. We also considered the airflow and air exchanges in the aircraft. The inside of an aircraft is obviously different from the inside of a hospital room. In our aircraft, the air exchange is 10 air exchanges per hour, but there's an additional flow of air from the back of the plane to the front of the plane. And finally, we wondered about ascent and descent, how those accelerative forces would affect the dispersion of particles. We found that aerosols were dispersed throughout the aircraft, even into the cockpit. We did find, however, that high flow was no worse than nasal prongs and BiPAP, and intubation were actually better. These are preliminary results, and we're conducting additional flights to verify our results, but this is of extreme importance to the safety of our crew. Many remote communities depend on using high flow and non-invasive ventilation techniques to buy time for their patients and to avoid mechanical ventilation. So it's absolutely important for us to be able to transport patients using these modes without the need for intubating and mechanical ventilation. At any time, The Canadian Armed Forces might undertake missions for the protection of Canada and Canadians anywhere in the world. The types of missions are unpredictable, and so the operational issues and health threats facing our soldiers can be diverse. Today, I present no answers to our current threat, COVID-19. I'm really hoping some of our later speakers, like Dr. Langley, will have some of the answers. Even so, I hope I've convinced you that EMS, hospitals, academics, civilian defense scientists, and military researchers all need to collaborate to solve this COVID-19 problem and future problems that we face as a military organization, as a country, and as a world. Thank you very much.
Of the current challenges facing both the Canadian Forces and the civilian areas of transporting infectious patients, and as you kindly pointed out, our research collaborations with our civilian partners really started under your watch during your time in the Canadian Forces, regular forces, and they all continue to this day. So, and I think that's the way forward, as Homer said, is the collaborations with our civilian partners. And now it's my great pleasure to introduce Dr. Joanne Langley. She is a professor at the Departments of Pediatrics and Community Health and Epidemiology in Dalhousie University. She's also the GlaxoSmithKline Chair in Pediatric Vaccinology at Dalhousie University, and she is currently the Canadian lead for the COVID-19 Canadian vaccine program. So it's my great pleasure to ask Joanne Langley to take the podium.
Good afternoon. My name is Joanne Langley. I'm a pediatric infectious disease specialist at the IWK Health Centre in Dalhousie University and a vaccine researcher at the Canadian Centre for Vaccinology. Thank you so much to the organizers for inviting me to be part of this symposium and for considering the role of vaccines in mitigating the current threat we're all living with. These are my disclosures. At the Canadian Centre for Vaccinology, we do research with government and non-governmental funding, as well as with industry. The funds for that go to Dalhousie University, where we do our studies. My objective is to provide an overview of current vaccine development and some updates on where the clinical research is at this point. So here's a dashboard from September 25th. The Johns Hopkins website, which I'm sure you're all aware of. So on the far left, you can see we've had over 32 million documented cases at this point. On the far right, almost 1 million deaths at this point. And the spread of disease is throughout the globe, 188 countries recording disease, but you can see it is denser in some areas. So a disparate burden of illness across the world. SARS-CoV-2 is a human coronavirus. This is a picture from the first coronavirus to really make the headlines, SARS-CoV-1 in 2003. But there have been these human cold viruses that have been circulating for some time. SARS-CoV-1 was followed by MERS, the Middle East virus that affected just one part of the world, but both of these had significant morbidity and mortality. Neither were as transmissible or caused a pandemic to the extent that SARS-CoV-2 is. So SARS is considered an emerging infectious disease, and these global health threats can either be emerging or re-emerging. Re-emerging infectious diseases arise because of changes in human behavior or of climate change, a number of factors. The point of this slide is just to say that there are many emerging and re-emerging infectious diseases. And as working or being affiliated with the military and being so outward looking as part of our international community, you would expect, no doubt, be aware of this. After SARS-1, the world decided that it really needed to move ahead these international health regulations that were conceptually in development, and they ultimately went into force in 2007. They're an international legal instrument that's binding on 194 countries, which are members of the World Health Organization, and outlines the minimum requirements for a functional public health system that allows countries to detect and respond to disease outbreaks in their communities. One of the aspects of the IHRs is the pandemic health threat of international concern, sometimes abbreviated to PHEIC. Since 2007, when this policy came into effect, we've had 7 declarations, 6 declarations, the most recent being SARS-CoV-2. How do we mitigate this threat? We, of course, are using public health measures, physical distancing, masks, contact tracing, and so on, infection prevention and control, which includes behavioral measures, engineering controls, and personal protective equipment, and key are vaccines. When we think about vaccines, we can think about passive immunization and active immunization. And one of the talks today will be on passive immunization, which is the transfer of exogenously produced antibodies into an individual. This can be by intramuscular route or intravenous or transplacental transfer of maternal antibody to the baby through the umbilical cord or through breast milk. We're going to focus on active immunization, which is introduction of a molecule recognized as foreign that leads to immunity. The ideal result of an adaptive immune response is that you will have a specific immune response that has memory. That is the ability to respond specifically to the antigen it was exposed to. So here you have that pictorially represented. Here's the antibody titer on the y-axis and the relative time. So when you get an infection or are exposed to a vaccine, your body has this first exposure and develops an immune response, and that will wane a bit over time. But when you're exposed to that antigen again, your response will be quantitatively greater, and it will be specific for that immune target, and that is called the secondary immune response, which is the goal of vaccines. So what vaccines do we have in development? There's 180, I think, of them that are in development at this point. Most of those are at very early stages, and as promising as they might be, are unlikely to deliver a vaccine that we could use in public health programs in the next year. The ones we are focusing most on are the ones that are already in phase 3 or have the possibility of being in phase 3 in the next year. These are 2 trackers I particularly like. This one is CBC, Emily Chung, and this one is the New York Times. You can see here that they track them according to phases. So what are those phases? Phase 1 is first in humans, generally small numbers, healthy people. Phase 2, we expand to other populations, such as older adults, people with maybe comorbidities, and we move from pure safety evaluation and is there immune response to looking at what does the dose of antigen have, what's its relationship to the quantity and quality of the immune response, how much antigen do we need to put in the vaccine, and so on. Phase 3 is the pivotal point where we look at efficacy. Does the vaccine protect people who get it? So which vaccines are farther ahead at this point? So here are the vaccines that are in phase 3 clinical trials. So this is where we have a randomized controlled double-blind process. We allocate people to a vaccine or placebo. The patient or the study staff don't know which vaccine they're getting. And then we send them out into the world and we follow them for illness. And we're trying to determine, does the vaccine protect them against illness? The Vaccines that are in phase 3 trials are the lipid nanoparticle vaccines, which deliver messenger mRNA. So that's Moderna and BioNTech, Pfizer, Fosun Pharma. This is a new prototype for vaccine. There are no currently approved vaccines that have this platform. The next is viral vector vaccines. These are all adenovirus, but different types of adenovirus. So that's Janssen, the CanSino, non-replicating, AstraZeneca, and the one that's being used in Russia. Subunit platform vaccines, such as Novavax, which just started phase 3 trials, are more familiar to us. Most of our routinely programs in public health are using a subunit protein approach. Then there's inactivated virus, and these are currently mainly the Chinese vaccines are in phase 3 trials, but there are other ones coming up in the rear that will also be inactivated virus. So here are the platforms that can be used theoretically for viral vaccines. So in the top left, we have live attenuated. So for example, measles, mumps, rubella, those are live attenuated vaccines that do not produce severe disease, but enable the immune system to respond. Then we have whole inactivated vaccines, split inactivated, synthetic peptides, virus-like particles. There are vaccines that are used in commonly used programs for this, such as the human papillomavirus vaccine. DNA or RNA, no authorized vaccines using this platform. Recombinant subunit vaccines, recombinant bacterial vectors, none use this process right now, and recombinant viral vectors. So here's the SARS-CoV-2 virus, a picture of it developed by graphic artists at CDC. The gray surface is the spherical envelope that surrounds the nucleus of the virus. The orange bits are transmembrane proteins, or M protein, they're structural. The yellow bits are also envelope proteins. The red spikes are the really important ones, we think. They're called S proteins, and they're what the virus uses to gain entry into the cell. And they also create this coronavirus effect. This is an electron micrograph of a human airway cell infection by SARS-CoV-2, and those little projections are epithelial cell cilia, which beat multiple times and get rid of mucus on our respiratory tract. This is a close-up of the spike protein, SARS-CoV-2 spike protein, which is the red projection on the left, and it is even more intricate. So it's all the RNA that's in the virus in a three-dimensional form, and part of this spike protein has a receptor binding domain for the ACE receptor here on a pneumocyte or in many epithelial cells in the human. And that's how the virus gains entry to the cell. So most of the vaccines focus on this spike protein because that's how the virus enters our cells. Now we're learning a lot about the pathogenesis of COVID-19 because of the way it enters. Just a word to say that in non-infectious times, the ACE protein is part of the renin-angiotensin-aldosterone system, which regulates our blood pressure, and it's on pneumocytes, but also, as I said, on various endothelial cells, and that is part of the research into the pathogenesis. So then on our next slide here, we see in blue the phase 3 trial vaccines. DNA vaccines have not entered phase 3 trials. Neither have the virus-like particles. The first subunit protein one to enter is Novavax, which started this week. So The World Health Organization has helped regulators around the world and governments by identifying a target product profile for vaccines to help developers understand what we really need in a vaccine. These are either critical or minimal characteristics. The target population should be all ages. There shouldn't be contraindications, because that will make it difficult to deliver and we won't be able to give it to the people who need it most. There should be a favorable benefit risk profile. Ideally, you'd like an efficacy of at least 70%, but the minimum is 50%. It would be preferred to have a single dose rather than multiple dose. It should protect for at least 1 year. And on a practical side, we have to be able to scale up production at cost, which will allow use in low and middle income countries. Now, the practical side is the biomanufacturing and the ability to scale vaccine production up for clinical trials, but also for use in public health programs. Operation Warp Speed, which is the entity in the US that has been tasked with finding vaccines for Americans, published these criteria that they are using to pick their front runners. And they are robust preclinical data or early stage clinical trial data supporting their potential for safety and efficacy. Second is potential to enter large phase 3 efficacy trials now, so July to November 2020, and deliver efficacy outcomes by the end of 2020 or the first half of 2021. And you see here acceleration support. So Operation Warp Speed is providing the funds to do many of these phase 3 trials, which cost hundreds of millions of dollars. And this financial barrier could prevent companies from going into this very risky process for a vaccine that might not work. So Operation Warp Speed de-risks the process by paying for the trials and prepaying for vaccine before they know that it's available. And this is the strategy that's being done around the world. The third criteria was that the prototype should be based on vaccine platform technologies that permit fast and effective manufacturing, and that the manufacturers can demonstrate that they can produce multiple doses of vaccine. Lastly, they chose certain vaccine platforms that they thought were most likely to yield this safe and effective vaccine. So that's the mRNA platform, replication defective live vector platform, recombinant subunit adjuvanted protein platform, or attenuated replicating viral vector platform. I'll just tell you for a few seconds about the Canadian Immunization Research Network that I'm involved in, CERN. This is a network of 150 investigators around Canada that does vaccine research to answer public health questions. It was originally formed to address influenza, but later was expanded. And we are a network of networks. So the Clinical Trials Network will be hopefully doing phase 1 and phase 2 trials of COVID vaccines. The other networks that are involved in kind of the spectrum of kinds of research that are needed to answer public health questions, such as social science questions, modeling and economic evaluation, serious outcomes related to hospitalization-based endpoints, and rapid safety surveillance are all also gearing up to answer COVID-related questions. The Clinical Trials Network has 10 sites, and many are experienced in early phase trials. We've also, as part of COVID, linked with other sites around the country and other networks, just in case we ever had to do a phase 3 trial and needed to enroll thousands of people. And we're collaborating with a number of small vaccine developers in early phase clinical trials. So when the phase 3 trials are done, we'll be at a certain point in what we call the vaccine life cycle. And this slide was given to me by Dr. Barbara Law, who's a safety expert in vaccines. So we can think about vaccine discovery, all the preclinical work that has to be done before the regulator in the country says, yes, you can do a clinical trial in humans. Then you do phase 1, 2, and 3 trials. At this point, the vaccine gets authorized for general use, but there's still many questions that we don't have an answer to, such as, what are the adverse events in large populations? Because we would have only tested it in 30,000, we may not know about more rare events. So it's very important that we also do phase 4 or post-marketing studies to look at these other effects. We also may not know if the vaccine provides indirect protection for people. So some vaccines, such as the pneumococcal vaccine, protect people who never got it because the vaccinees themselves have less carriage of pneumococcus in their throat. So that is an indirect effect of vaccination. In Canada, after the regulator approves vaccine, the National Advisory Committee on Immunization will make prioritization recommendations, and the Canadian Immunization Committee, which is a federal, provincial, territorial committee, will be involved in designing the implementation process and the rollout of these vaccines. And throughout this process, there are many things to consider besides burden of disease and vaccine characteristics, such as ethical and equity considerations, conformity of programs, cost effectiveness, and so on. So I'll pause there. Canada is part of a global process to procure vaccines. We're procuring them for our own citizens, but also through the COVAX facility, which is a World Health Organization and multiple country collaboration to procure vaccines for low and middle income countries, as well as for our own citizens. And this is accelerating vaccine development as well as procuring supply. And I would be most happy to answer any questions you may have.
Well, thank you so much, Joanne. That was an excellent talk and really highlighted all the challenges that go into making a vaccine for an emerging virus. Just remind all the participants, if you do have questions, just enter them in the YouTube chat box, and we'll be happy to answer them. But just to start off, I've got a couple questions for you, Dr. Langley. The first question I have for you is, as this is a military audience, and the vaccine will be rolled out probably to certain populations first, such as vulnerable populations and healthcare workers. And where would you see the military falling in lines with the getting the new COVID vaccine when there's a licensed vaccine?
Hello. So the question is, where would the military fall in line with whatever are the approved vaccines?
Mm-hmm.
Yeah, so we don't know for sure at this point which vaccines will ultimately be part of our programs. And that knowing which ones will be part of the programs is essential, because some of them have very particular handling requirements, cold chain requirements. For example, the messenger RNA vaccines, the Pfizer one, for example, has to be at -60. That will affect your planning for who to deliver that to. Also, the amount of vaccine that Canada will get from each vaccine manufacturer may change. So the first, rule suggests that the highest priority people to get vaccine are those who are primary care, frontline providers. And likely a number of people in the military would fall into that category, or who are high risk. Then there may be people within the military that would fall into that. So there may be different vaccines recommended for different groups, depending on early availability. For example, say we can get it in the first quarter of 2021. Those you would want to provide to your healthcare providers pretty soon after that with whatever vaccine is available. So I think I know that there is a military representative on the National Advisory Committee on Immunization, and those factors would definitely be considered as a special population.
Well, thanks, Joanne. And just while we're waiting for some of the other questions to come, we do have one question coming up, and is how can the influenza immunization campaign this fall impact the proliferation of COVID-19?
Yes, so influenza is a very important cause of respiratory disease and exacerbates a number of underlying chronic conditions like heart disease, lung disease, and annual immunization is recommended in many jurisdictions. So you will notice if you look on your local public health description of what is a screening criteria for a COVID test, that it's quite non-specific. It can really be any respiratory complaint. So that means that respiratory complaint could be, in fact, influenza or some other respiratory virus. If we increase the number of people that get their influenza vaccine this year, we will make it much easier for our healthcare systems, our ambulatory settings, our workplaces to be able to handle respiratory illnesses, because we will decrease the incidence of these respiratory illnesses. So I very much encourage people to get their influenza vaccine. There's no knowledge so far that there's any interaction between getting the two vaccines or that it's more harmful to get the influenza vaccine and then be exposed to COVID. The viruses are independent to our knowledge, and so the recommendation is to get your influenza vaccine, and it's probably even more important this year to get your influenza vaccine.
Well, thanks, Joanne. Leading on to that is there's a lot of talk about herd immunity and what that would mean for a COVID-19 vaccine. You know, what percent of the population would you say would need to be vaccinated for the so-called herd immunity with a vaccine efficacy of about 70%?
Mm-hmm. So herd immunity is this idea that a population can protect people that aren't vaccinated just because a certain percentage of people in the general population have immunity. So the population itself in some ways protects the whole, its members. And the highest way to increase herd immunity is if the vaccine itself provides indirect protection. So I mentioned that when I was speaking, in that some vaccines will reduce the likelihood of you spreading the infection if you're vaccinated. And many vaccines will not. They only provide direct protection to you. For example, the tetanus vaccine only protects you. It doesn't protect anyone else. If they're exposed in a combat to dirt, they need to have their own protection against tetanus. We don't know yet if this vaccine will provide indirect protection. And therefore, it's probably pretty likely that we'll have to have fairly high levels of folks being vaccinated in the general community to provide this kind of overall population protection, this herd immunity. So I guess the short answer is we don't know for sure. It will be very important that uptake is high, and we will study whether there's indirect protection as part of phase 4 post-marketing rollout of programs. So the estimates are really modeling estimates. With this level of efficacy, we might expect this level, with a certain level of efficacy of the vaccine, we might expect a certain level of indirect protection conferring herd immunity. It's a bit of an unknown at this point.
Great, and another question from the audience is how are sex and age expected to impact the vaccine's efficacy and risks?
Mm-hmm. So there is certainly a difference in sex, biological sex, we know, and the incidence of certain infectious diseases and the morbidity and mortality from it. Sometimes it's very hard to dissect the influence of this risk factor from socioeconomic considerations. And I think my view would be there's a lot we have to know yet. In the trials themselves, they are trying to balance enrollment so that there is a good distribution of sex and gender and socioeconomic considerations and racialized communities. But early on, it may be that the predominant information will be from healthy people, and whatever people enrolled. As trials roll out, again, I think this will be something that will be clearer as we roll them out into programs.
And another question is here is, sorry, I just it's coming up on the screen. How many more than 30,000 people studying in phase 3 would you like to see in safety and efficacy studies?
Mm-hmm. So this 30,000 number is something we hear about in the news because most of the trials are designed to determine a 50% efficacy, so 50% reduction in the incidence of disease in people who received vaccine compared to those who received placebo, because that's the minimum criteria in for many regulators. Some may have a higher efficacy point, like up to 70%, in which case you might need more people or a higher incidence of disease. So it's not entirely clear what will happen at the point where trials meet this base criteria for unblinding and analyzing their data. If they unblind and analyze at the 30,000 enrollment point, and it's shown to reduce disease by 50%, then they could apply for authorization in countries and start to roll out vaccine programs. Or regulators could say, no, we want to continue trials and see if you meet the 70% efficacy standpoint. Or we want you to enroll certain populations that are particularly important for our country. Those are the kinds of decisions that regulatory authorities might make, or the companies could decide on their own to make these decisions to continue enrollment. Some of the considerations that will have to be weighed at that point will be, is it more important to start rolling out a vaccine program with 50% reduction in disease? What will that mean for my country? As opposed to continuing on to get more refined endpoints. These are not just scientific decisions, they're ethical decisions and require a lot of input from folks other than just the scientists, I think. And so they could be answered perhaps with a less strong research design by doing observational studies once you implement programs. So those I think are some of the considerations. I don't have any, as you can see, absolutely definitive answers to those kinds of questions.
Well, thank you very much, Dr. Langley. Those were great answers and a very good talk and overview of the Canada's approach to getting a COVID-19 vaccine, which we're all waiting for with bated breath, and so we can go back to regular operations and regular life in Canada. So that was very enlightening. So thank you very much. And now it's my pleasure to introduce the next speaker. I'd like to introduce Michelle Scholzberg, who's associate scientist here in Toronto at the Li Ka-shing Knowledge Institute. And she's also now the new chief of her department. She's a hematologist and oncologist and leads the clinical research group here, and she's really an expert in hemostasis, and what she's been working on lately is a therapeutic for the very sickest COVID-19 patients that we're seeing in our ICU here. And I'll leave it over to Michelle now. Thanks very much, Michelle.
Hi, everyone. It's a great pleasure to speak with you all today. My name is Michelle Scholzberg. I'm a hematologist and clinician investigator at St. Michael's Hospital and the University of Toronto, and my focus is clinical trials. And I'm going to start my presentation now. I'll start by sharing my screen. Okay. So today I'm going to speak about COVID coagulopathy and our clinical trial that is addressing this very important and relevant clinical entity. So our clinical trial is called Rapid COVID Coag, or the RAPID trial. So hospitalized patients with COVID-19 appear to have laboratory findings that are suggestive of coagulopathy. And up to 70%, in fact, of hospitalized patients with COVID-19 have some indicator on their lab testing that coagulopathy is present. So this is a really high proportion of individuals that appear to be affected. And elevation in D-dimer is what appears to characterize the coagulopathy. Very interestingly is that this elevation in D-dimer seems to correlate with the need for critical care and death. So from this, it became clear to many scientists and physicians and other healthcare workers around the world that thromboinflammation is a key pathophysiologic mechanism of severe COVID-19. And this is based on cohort studies, the majority of which are retrospective, indicating that up to 50% of individuals who are admitted to intensive care units are diagnosed with thromboembolism. And we think that patients with COVID-19 progress through various stages of thromboinflammation. And we also think that it is important to intervene early in this thromboinflammatory pathway to hopefully diminish the risk of poor outcomes, including mortality. And this hypothesis was put forward initially in late February, early March by Tang et al., who were the first to report that there seemed to be a lower 28-day mortality amongst individuals who received heparin thrombotic prophylaxis in Wuhan compared to those who did not. And this seemed to be particularly true of individuals who had markedly elevated D-dimer. And so why are we interested in heparin, which is one of the most commonly used anticoagulants and the most commonly used anticoagulant in hospital? Well, we're interested in it for its anticoagulant effect, but our interest actually extends beyond that. And that is because there are numerous animal models that indicate that animals with acute lung injury and sepsis appear to benefit from heparin, as it seems to diminish the risk of capillary leak and improve endothelial function. And these animals seem to have an improvement in their acute lung injury when treated with heparin. Additionally, heparin may bind directly to the SARS-CoV-2 spike protein, interfering with viral attachment and entry. Thus, heparin itself might have antiviral properties. And also, it has long been known that heparins have anti-inflammatory effects, but this has also been shown, in fact, in patients with COVID-19. And so there are decreases in IL-6 levels in individuals treated with low molecular weight heparin. So evidence is urgently needed. So because this coagulopathy is becoming increasingly recognized as a manifestation of severe COVID-19, the International Society on Thrombosis and Hemostasis fairly recently published a guidance document on its management, but that is despite the current paucity of evidence. And so really, unfortunately, clinicians, just like every other human on this earth, has been responding emotionally to the pandemic. But unfortunately, this has led to many institutional protocols recommending therapeutic anticoagulation with heparin-based therapies, despite the lack of prospective evidence. And this is dangerous because we just don't know if it's safe. So what is the current landscape for anticoagulant intervention trials in the setting of patients with COVID-19? Well, we recently published in the Journal of Thrombosis and Hemostasis a scoping review looking at what clinical trials exist right now. And in fact, there are 20 clinical trials that are ongoing in this realm. And I'm circling right over here, our trial, which is the Rapid COVID Coag. So there are many people that have had a similar idea to us, but all of these trials use slightly different methodology, and I'll justify and explain why we chose our methodology. And so what's interesting is that some of these clinical trials include patients that are in the ICU as well as not in the ICU. Most of these clinical trials predicate eligibility based on D-dimer values. And the majority of these clinical trials include really important clinical outcomes such as death as the primary outcome. And so this is just showing all of these trials. And so now I'm going to talk about Rapid COVID Coag, which is a Canadian-led international trial in this space. And so I am one of 3 principal investigators on this trial. I'm also joined by Professor Peter Uni and Mary Cushman. Peter Uni also works with me at St. Michael's Hospital and at the Lee Cushing Knowledge Institute. He is one of the most world-renowned clinical epidemiologists and clinical trialists, in fact, one of the most highly cited clinical epidemiologists, and is a Tier 1 Canada Research Chair. And Mary Cushman is also an incredibly eminent epidemiologist with a focus on thromboembolism. She's out of the University of Vermont Medical Center that is also home to a very large biorepository. But you can see here from our steering committee members that we have individuals across Canada and across the world with really outstanding expertise, both from a clinical trials methodology perspective, statistical analyses perspective, and also from a content perspective. And so we have real giants in thrombosis and hemostasis. And so I'm sure you will recognize many names here. And so we're really fortunate. So this trial is a 2-arm, parallel, pragmatic, rapid, multi-center, open-label RCT. And so the emphasis for this trial is its rapidity, because scientists need to move as quickly as possible to develop sound clinical evidence to help guide clinicians on the front lines. And we feel very strongly that this is our responsibility. And so this trial is also designed as being pragmatic, meaning that it's practical, it's easy to implement, because it is completely unethical to overburden clinicians in this time. And so, as I said, a 1-to-1 randomization, so a flip of a coin using a web-based central randomization system that will either randomize patients to standard care, which is our usual dose of heparin, unfractionated and low molecular weight, compared to therapeutic anticoagulation, again, pragmatically defined using either unfractionated heparin or low molecular weight heparin, which are the doses that we typically use to treat thromboembolic events. And our primary composite outcome is that of ICU admission, non-invasive positive pressure ventilation, so CPAP and BiPAP, as well as invasive mechanical ventilation or death at 28 days. And just to re-emphasize, our goal is to start heparin early with the idea that we think that this will decrease pulmonary inflammation and downstream sequelae, including death. So to be eligible for our clinical trial, patients have a 72-hour window, meaning that these are individuals who have to have confirmed COVID-19 via PCR, and they need to be admitted to hospitals specifically for COVID-19. And so this is not a clinical trial of patients who are, let's say, admitted to hospital for an elective surgery and happen to be found to have positive COVID-19 testing. No, this is for patients who are admitted because they're symptomatic. And also these individuals have to have a positive D-dimer, and we've defined this in 2 ways. Either as being above upper limit of normal with the presence of hypoxia, meaning an oxygen saturation of less than or equal to 93% on room air, or a D-dimer of greater than or equal to 2 times the upper limit of normal. This is obviously also a study of adults, and we're looking for patients to either be able to give informed consent themselves or for a substitute decision maker to be able to provide consent for them. And we have excluded individuals where the risk of bleeding would be deemed unacceptable, or where the risk of thrombosis is in fact stimulated by other important variables. So pregnant patients, for example, are excluded from this clinical trial. Also individuals with significant anemia where their risk of transfusion is higher, or they have significant thrombocytopenia, low fibrinogen, a high INR. We're also excluding patients who are already on intermediate dosing of low molecular weight heparin, where the most responsible physician thinks that the dose cannot be changed. Individuals who are already on therapeutic anticoagulation at the time of screening, meaning people with another indication to be on a blood thinner, are excluded from the trial. Patients on dual antiplatelet therapy when one of the agents cannot be stopped safely. Patients who have a higher risk of bleeding otherwise, so patients who've been admitted to hospital in the last month, known for a bleeding disorder, or a known history of heparin-induced thrombocytopenia or allergy. And also we're excluding individuals who've already met a component of our primary outcome at the time of screening. So that means that individuals that are already admitted to the ICU, patients who already are on CPAP or BiPAP or already intubated are not eligible for the trial. Also, we're excluding patients where the most responsible physician thinks that they're imminently dying. If that's the case, it's also not ethical to include them in the trial. Really importantly, co-enrollment in another COVID-19 related clinical trial in general is not an exclusion criterion, as we felt very strongly, as have many clinical trialists in this space, felt that as the standard of care is shifting and we all need to ensure that patients have adequate opportunities to engage in clinical trials, that we didn't want to exclude other opportunities for patients. But of course, patients who are enrolled in another antithrombotic trial would be excluded. So I've already talked about our primary outcome. Our key secondary outcomes are things that people would generally be interested when we're providing anticoagulants. So we're interested in bleeding risk and clotting risk. And so we've defined major bleeding as per the ISTH definitions. We've also included blood transfusions, organ support indicators, as well as heparin-induced thrombocytopenia. We're also including a biorepository component to the clinical trial. And so we're interested in the trajectories of different inflammatory and thrombotic biomarkers, and certainly interested in collaborating in that regard. As per our sample size, we need 462 patients, so 231 per group, to detect a 15% risk difference from 50% in the control group to 35% in the experimental group. So many people can indicate that, is this really all the patients that you need? Are you sure you don't need thousands? The answer is no, we don't think that we need thousands. And we don't think so, and this is based on personal communication and exchange with numerous investigators in Wuhan, China, who provided data for our team so that we could help refine our sample size as much as possible. And this is to account for a power of 90% and two-sided alpha of 0.05. Importantly, though, we are moving forward with humility in that we recognize that we could be wrong. Maybe we need far more patients, maybe we need far less. And so our analyses are also adaptive, both for efficacy, but also include conditional power analyses so that we can make adjustments along the way. It's possible that we could end the trial early, either on the basis of futility or if we have a signal that, let's say, the therapeutic arm really is much more efficacious. Our statistical methods were deliberately very straightforward, and we did this with a frequentist approach. And so it's really very simple. It's a chi-square test for the main analysis of the primary outcome. This was very, very deliberate because this will give us the answer as quickly as possible. Certainly, we're going to include multivariable analyses to allow us to adjust for relevant covariates that include age, sex, cardiovascular disease, level of D-dimer and hypoxia, but also really importantly, race and ethnicity, as it has been shown in numerous reports that individuals of African or Hispanic descent appear to be at higher risk of death from COVID-19. Now, some of that can be explained by social determinants of health, but we think that there's also a biological explanation for this, as we've also recently found that individuals of African and Hispanic descent have higher baseline thromboinflammatory markers, including D-dimer. So to that point, we really feel very strongly that developing a biorepository in conjunction with our clinical trial is of paramount importance, because we are desperate around the world to understand this thromboinflammatory state that seems to characterize COVID-19 more so than other viral infections. And so these are just some of the markers that we are interested in measuring, and we are very interested in collaborating. So here's our organizational chart. And so our protocol is developed, our data is coordinated, managed, and analyzed, and all of our regulatory related work for the Canadian sites and international sites are managed through St. Michael's Hospital Li Ka-shing Knowledge Institute, led by myself and Dr. Peter Uni. The biorepository is going to be housed at the University of Vermont, but in close collaboration with Doctors Paula James and David Lillicrap, who've in fact already secured funding for von Willebrand factor related studies. And so this will allow us to collect samples, to process them, and then analyze them and collaborate in the future with other individuals. And we just wanted to indicate that we are very interested in collaborating with the DRDC. And we've already had some interactions with Dr. Rind in this regard. Our US sites are managed from a regulatory and administrative perspective by Versity, led by Lisa Baumann-Kreutzer. Versity is the new corporate name of what used to be the Blood Centers of Wisconsin. In Europe, it's led by Dr. Fanula Nienli at the University College Dublin. In Brazil, it's led by Dr. Elnara Negri in Sao Paulo, Brazil. And we are also moving into the Middle East with Dr. Musad Al-Hamza and Dr. Moza Al-Marshudi, who are leading our teams there. And we are hoping that we're going to move into Serbia as well. Finally, we've also moved into Argentina with numerous colleagues there. So it's really remarkable how much an international interest and collaboration and honestly pure scientific spirit that exists around the world. And we're really delighted with the team that we have. So here are our Canadian sites. We have 19. We're opening them in very rapid succession. And our rapid international, which of course includes our Canadian sites, but also some select sites in the United States, Brazil, Argentina, Ireland, Serbia, Saudi Arabia, and UAE, as I said. Our milestones to date. And so we were unfortunately unsuccessful in our CIHR submission for the COVID-19 related competition. There were 339 submissions, 30 were funded, and we were ranked 31st. So it was a bit of a blow for us, but it also speaks to how highly regarded our clinical trial was at CIHR. And so thus far, we are funded by DRDC Task 54, by our hospital foundation, by St. Michael's Hospital Foundation, by the Ottawa Hospital Competition, Michael Garron Hospital Competition, and the Southeastern Ontario Academic Medical Organization Competition, and an International Invent Kickstarter Award. So despite us not achieving at CIHR, we right now have enough seed funding to keep us going, and I keep applying for additional grants. Right now, we're open to recruitment at 3 centers, but we're moving really quickly. In fact, we have 2 site initiation visits occurring right now as I'm speaking to you. And despite only being open at 3 centers, we've recruited 44 participants thus far. We have contracts fully executed in Saudi Arabia and the US, and so those SIVs are occurring right now. As soon as those SIVs occurred, we just launched at all the other hospitals that are connected at those sites. And we have 5 Ontarian sites that have completed their SIVs. And so it's literally a matter of hours to days before we're launched at other sites. So we believe that this trial has the potential to immediately impact the clinical care of patients with severe COVID-19 internationally, irrespective of our findings. And so in the midst of this pandemic and the secondary healthcare crisis, it's time now more than ever for randomized care of agents with sound biological plausibility rather than haphazard best guess care. And we also feel strongly that this is a well understood therapy with predictable side effects. And we know that this will undoubtedly lead to subsequent patients receiving evidence-based therapies and spared from unnecessary harm if, for example, we find that we're wrong and that therapeutic anticoagulation is not needed. As I've emphasized throughout my talk, we're really interested in collaborating. And so St. Michael's Hospital, as you know, is a large Canadian level 1 trauma center led by Dr. Andrew Beckett. And I'm very fortunate to be able to collaborate with him. We have a profound interest and expertise in coagulopathy associated with trauma. And so my focus in hematology is that very thing. It's hemostasis. So my expertise is bleeding and clotting, and my entire research program surrounds that. I also believe that we have the ability to deliver from a content expertise perspective, from a research methodology and infrastructure perspective. And I hope that the RAPID trial will be an initial step towards a fruitful academic collaboration and program development between our two groups. And so I wanted to thank you all very much for your attention, and I'd be happy to take any questions now. Thank you.
Thanks for that great review of your trial, Michelle. And just a reminder to all the audience, you can start typing in your questions into the YouTube chat box, and we'll be happy to answer them. But while we're waiting for some questions to come up, a couple questions that I had for you is, now in the ICU, there's been some demonstrated improvements in supportive care with things like tolerating happy hypoxia, proning the patient, and even the use of steroids. Now, how do you think, with all these improvements, do you think your trial may be overshadowed by some of these other improvements, and you may have to do a larger sample size because it's going to be harder to tell what's going on with all these other new clinical protocols coming into place?
Thanks so much. That's a great question. So you're absolutely right that the standard of care for COVID-19 is a moving target. And so that's why it's so important that we incorporated the conditional power analyses, because we assumed over the course of the clinical trial that things were going to change. And so we don't know. We don't know. Once we do our conditional power analyses at 25 and 75%, that will tell us the answer. We still feel that it's likely that we're going to see a pretty significant signal. And so we're hopeful that therapeutic anticoagulation will prove to be of some benefit. Interestingly, though, we think that there's probably a really important distinction between patients who are admitted to the medical wards versus those who are in the intensive care units. And so we think that there needs to be a differential approach taken to the ICU patients. And so essentially when patients that are enrolled in the RAPID trial get admitted to the ICU, we back away from those patients and allow the most responsible physician to adjust their care as they deem fit, because essentially most of these patients are being admitted to the ICU because they're developing, progressive hypoxic respiratory failure. And so top in that differential includes a pulmonary embolism. And so if they think that they need to, therapeutically anticoagulate the patient, let's say if they were randomized previously to the standard care arm, then they are absolutely free to do so. And also because we felt strongly that those patients should have the opportunity to enroll in other clinical trials that might have a more aggressive approach with regards to antithrombotic therapy. And so I think the honest answer is I don't know, but that's why it's important to include these conditional power analyses and sort of see, how things go along the way.
Very good, thank you. And a question from Dr. Dana Devine is, does the platelet level fall before D-dimer levels go up? Is there a platelet contribution, for example, factor 13 A chain release, loss of plasminogen activator inhibitor? And do you anticipate the effects of low molecular weight heparin on complement activation?
Such a great question. Thank you so much. So it's a great question about the platelet count fall. What's really been unique about the coagulopathy that seems to characterize COVID-19 is that thrombocytopenia does not really seem to be a consistent finding. Some patients do have mild thrombocytopenia, but not to the extent that we are accustomed to seeing in patients who are admitted to hospital with a viral infection. And so as hematologists, we were all sort of getting ready to sort of see a bunch of patients with thrombocytopenia or low platelet count when they'd be admitted with COVID-19, and we are not seeing this? And so the answer is that typically no, the platelet count does not seem to fall in relation to the D-dimer level. But when the platelet count does fall, it seems to again associate with a poorer prognosis. And so as the coagulopathy progresses and patients declare themselves at the end of the line with disseminated intravascular coagulation, their fibrinogen falls and their INR increases, though that's indicating that the patient is really becoming systemically very unwell. And so at that point, they do follow a much poorer course. So great question about, is there platelet contribution? There's lots of people who think that the platelets are contributing. There's people who think that the neutrophils are contributing. There are people who think that the fibrinolytic system is contributing. And there are many who also think the von Willebrand factor is contributing. And so the answer is likely everything is contributing to some degree. I think there's really a lot of interest in von Willebrand factor in particular right now, because these patients on autopsy studies seem to have evidence of microvascular thromboses in the lungs, akin to what we would see with a thrombotic microangiopathy like TTP, or thrombotic thrombocytopenic purpura, that we know is mediated by excessive von Willebrand factor. And we found in earlier studies that these patients have really, really high von Willebrand factor levels and really high factor VIII levels as markers of endotheliopathy. So the verdict is out. And I think that all of your hypotheses are really valid, which is why we really need to do translational work.
Okay, we have time for one last question, and the question from the audience is, what characteristics would an ideal anticoagulant for COVID-19 have?
Great question. So an ideal anticoagulant would be one that would have anti-inflammatory properties. So as Dr. Beckett was just saying, he said that we know that there's evidence that corticosteroids seem to improve prognoses in patients with COVID-19. And so diminishing inflammation seems to be important, which is why we're intrigued by the heparins. But that said, we don't know the answer. Are the heparins better than an oral anticoagulant, such as the direct oral anticoagulants? We don't know the answer to that. But it seems that right now, most people are interested in an anticoagulant that also has an anti-inflammatory effect. Other things to consider is you want to consider end organ damage. And so as patients become progressively ill, you want to consider whether or not their kidney function and liver function is becoming problematic in their ability to be administered anticoagulants. So that's another reason why the heparin class of drugs is one that we went with, because low molecular weight heparin and unfractionated heparin are ones that you can actually administer even in the face of significant end organ damage. So that's why we chose those.
Well, thanks very much, Michelle, and great talk and great answers to questions. And now we're going to move on to Dr. Dana Devine's talk. Dr. Dana Devine, she's the Chief Medical and Scientific Officer at Canadian Blood Services. She's a professor of pathology at UBC, and she's the founding member for the Center for Blood Research in BC. So it's my great pleasure to introduce her talk, and I've worked with Dana extensively. So she's going to talk on convalescent plasma, and I can say that the Canadian group here, they were working on convalescent plasma before Donald Trump even mentioned it. So it's my great pleasure to give the floor to Dr. Dana Devine.
Hello, my name is Dana Devine. I'm the Chief Scientist at Canadian Blood Services and a Professor of Pathology and Laboratory Medicine at the University of British Columbia. Today, I'd like to share some information with you about Canadian Blood Services and the work that we're doing toward a potential treatment for COVID-19. First of all, let me just make a couple of disclosures so that you all know that I do have some affiliations with industry, but none of these are relevant to the topic that I'm going to cover today, but I just did want to declare them. What I'd like to talk about today is something called convalescent plasma. And the idea behind convalescent plasma is that when we get infected, any kind of infection can cause the body to mount an immune defense response. And the usual way that our body does this is to make antibody molecules that attack the invading pathogen. So that could be a virus, it could be a bacterial infection, it could be a parasite. And these antibody molecules really are our primary defense against that infection. So they help signal the destruction of invading pathogens in our body. Because antibodies actually float around in the plasma of blood, you can actually collect them from blood donors. And these antibodies can be given to patients who are still sick with a particular disease. And the idea of transferring antibody molecules that can attack a pathogen into somebody who is sick with that same pathogen is a process called passive immunity. So you're giving someone else's immune system to someone who is ill. This idea of passive immunity has been raised as a potential strategy to help patients who have COVID-19 disease and who have not yet made an adequate immune response to helping them get rid of the virus. So what we're interested in is understanding whether convalescent plasma, or the plasma from people who have recovered from COVID-19, is actually a good source of antibodies that can neutralize the virus that causes COVID-19 disease. In other words, can we use this kind of convalescent plasma as a passive immune strategy to treat COVID-19? So first of all, we have to realize that convalescent plasma therapy is not a new idea. The photographs that are shown here are pictures from the Spanish influenza outbreak in 1918. So the idea of transferring the antibodies from someone who's recovered into someone who's still ill has been around with us for well over 100 years. And a group has recently gone back and looked in the ancient information in medical libraries of papers published around that time. And they could show that if you looked at all of these papers, that the number of people who died from the disease was reduced if you gave them the convalescent plasma of people who had recovered from influenza. So you essentially dropped the fatality rate in half. And they also were able to show that the fatality rate was lower if you gave the treatment earlier. So these are sort of two of the fundamental questions with the use of convalescent plasma, is when do you give it, and whether or not you can actually provide evidence that it really works. So let's jump a century and look at the convalescent plasma treatment for coronaviruses. So this is the kind of virus that causes COVID-19. And we do have a little hint that this might be something worth looking at. And we have that hint from the SARS outbreak in 2003. So in that outbreak, there were about 80 patients who were all healthcare workers in hospitals in Hong Kong, where they had quite a severe SARS outbreak. And these people received convalescent plasma. And then they were assessed at 22 days after their onset of symptoms as to whether they'd had a good outcome, meaning they essentially were discharged from the hospital, or whether they'd had a poor outcome, meaning they were either still hospitalized or they had died. The convalescent plasma appeared to improve the outcome if you were in the younger age group. So you can see in the red boxes that you get a good outcome was happening more frequently in younger people than in older people. And similarly, if you got treated earlier in your disease course. So the idea here is that you're giving antibodies to patients who either don't have very good ones yet or don't have enough of them. So this was an interesting hint that perhaps convalescent plasma could be used to treat coronaviruses. So if we fast forward to what's going on in 2020, we're really interested in understanding if, given this history of the use of convalescent plasma in other viral illnesses, could this actually be a useful therapy for COVID-19? There's a few hints in some early studies, people who had used convalescent plasma in treating individual patients with no control group. And also some of those studies reported that you could see improvement in patients, and then others reported that there wasn't any effect. And that again seems to relate to how soon you got patients treated, and that the convalescent plasma could have much less impact in people who were seriously ill. So how does this whole thing work? Well, this is a little schematic showing how the coronavirus gets inside cells. And you'll see on the left-hand side that there's a drawing of a virus here, and all these little blue spikes that are sticking up off the virus is what gives the coronavirus its name. So that supposedly looked like a crown to someone who first visualized this virus. What those little blue things that are sticking off the virus will do is they bind to a specific receptor on the host cell, and this is called an ACE2 receptor. And once the virus is bound to that receptor, the cell internalizes the virus, and then the virus's replication RNA will take over the host cell and start making lots and lots of viruses. So the virus will replicate in the cell once it gets inside. So the idea behind convalescent plasma is that if you have antibodies, the antibodies should be able to bind to the virus and block it from interacting with the receptor and preventing the uptake of the virus into the cell, and therefore the cell never becomes infected. So the idea here is that if you have these so-called neutralizing antibodies, in other words, they're neutralizing the virus, the more of these antibodies you have, you probably are going to have a better chance of getting the virus out of your system and preventing the virus from moving from one cell to another in its replication cycle. So we're very interested in donors who have high levels of antibody because we think that's probably going to be more effective for recipients. And we are testing all donations for the antibody level. So one of the other questions that comes up in these kinds of studies is a plasma transfusion still has all the risks associated with any transfusion. And so we also worry about an additional phenomenon that occurs because you're giving antibody against a virus to people who are quite sick. And there's some evidence in the medical literature that you can sometimes cause something called antibody-dependent enhancement of a viral illness. And this essentially means that when you give the antibodies from someone who's recovered to someone who's sick, you can really get that sick person's immune system over revved up, and that can actually cause additional severity of illness. And so there is some evidence in the literature which actually looked at this question. There's a very large set of data in the United States from about 20,000 people who received convalescent plasma through an emergency investigational new drug protocol. And when these 20,000 recipients were analyzed for adverse events from the transfusion, there was no evidence that you had this antibody-dependent enhancement. So that was very good news. It looked like the use of convalescent plasma to treat COVID-19 was going to be safe. So what's going on in Canada? Well, in Canada, we have 4 clinical trials that are underway, or 4 types of clinical trials that are going. The first is up and running. It's called Concord 1. This is a randomized controlled trial of convalescent plasma in patients who have just appeared at the hospital with breathing difficulties, and they get treated within 24 hours of admission. It's intended to be a large study. We need about 1,200 patients in the study to complete it. And these patients are randomized. 2 patients will get convalescent plasma for every 1 patient that gets the regular standard of care. The primary endpoint for the study is whether these patients end up on a ventilator or die within the first 30 days. And the dosing of the convalescent plasma is a single dose of 500 milliliters. There's a similar study going on in children, which is a much smaller study because, as you probably know, this COVID is much less common in kids, or at least kids don't seem to get sick with it. And so this is a study being run out of Sick Kids in Toronto that's about a 200-patient study. And the primary endpoint here is a little bit different because children don't seem to die of this disease particularly. And this is about decreasing the time to seeing a clinical improvement in the child who's treated. The third study is called REMAP-CAP, and this is actually a large international trial group, a group of investigators who do these kinds of trials, and they're focused on people who have very severe pulmonary disease. So this is a group of investigators who were studying new drugs to treat influenza and that sort of thing. And they have added an arm of convalescent plasma therapy to their ongoing international trials. So they're targeting a different patient group. They're actually looking at patients who are very severely ill and in the intensive care unit. So these are people who are probably already on ventilators, they're not doing well, and they're looking at convalescent plasma as essentially a treatment of last resort. The initial studies that are in the literature looking at this group of patients seem to show that it's probably not a very effective therapy, but we don't have a really properly designed study to look at that. And then in Canada, we also have the ability for a physician to open a clinical trial to treat a single patient. So this would be patients who are not eligible for one of the other trials or might be in a hospital that's not participating in the clinical trials. And a doctor can actually ask Health Canada for permission to have a clinical trials license just to treat that one patient. So far, we haven't really seen this mechanism being used, and patients are being put into the larger clinical trials. So where does all this convalescent plasma come from? Well, it comes from our fellow Canadians who are willing to undergo a procedure called plasmapheresis, where we essentially hook you up to a machine that collects your blood. We remove the plasma and give you back your red blood cells and your platelets and your white blood cells. So for the actual eligibility for convalescent plasma donors in Canada, people who have either had COVID-19 and it's been confirmed by a laboratory test, or are what we refer to as presumptive positive, meaning, for example, the person that you live with had a positive test and you were sick at the same time. Those folks can register at the Canadian Blood Services website, which is simply blood.ca. These donors must meet all the regular qualifications to be a plasma donor. So we want to make sure that the plasma meets all the same safety standards that regular plasma collections do. So at the moment, we're asking for donors who are less than 67 years of age and that they satisfy all of the regular blood donor questionnaire questions. The plasmapheresis procedure that returns all these cells to the donor actually harvests between 500 and 750 mils of plasma in each donation. Initially, we've been seeking male donors and female donors who've never been pregnant because we worry about a possible complication of plasma transfusion that's called transfusion-related acute lung injury. And obviously, because this product is being given to patients who have COVID-19, we don't want to put them at risk of doing anything that's going to cause further lung injury. All the donations that we provide for the clinical trial have their antibody titers measured by our National Microbiology Lab in Winnipeg. We make sure that the antibody titer, or the amount of antibody in that plasma, is fairly high, and this is a little bit esoteric, but for those of you who understand this business, the antibody titer that we use is at least 1 in 160. And then if we have a titer that's that high, we release these units for the clinical trials. If a donor donates a unit that doesn't have that high level of antibody in it, we still keep those units and we use them for the production of testing panels for laboratories around the country that are doing convalescent plasma testing or that are testing general population to look at the incidence of how many of us have actually had COVID-19. So where are we now? Well, the Concord 1 study, so this is the patients who are just coming down with COVID-19. There are about 40 hospitals across the country that have started up this trial already. We've randomized 105 patients as of mid-September, and a lot of convalescent plasma has been collected by both Hema Quebec, our sister organization who runs the blood service in Quebec, and Canadian Blood Services, which provides blood and blood products to the rest of the country. The Concord Kids, so the pediatric equivalent study, has been launched, but has not got very many patients into the study because there really haven't been very many sick kids, which is actually a good problem to have. The REMAP-CAP study of the very ill patients has not yet launched. So the challenge that we're looking at is these are very large studies, and the number of cases that require hospitalization in Canada is relatively low right now, which is very good news for our hospitals, but maybe not such great news for these kinds of clinical trials. So recruitment is very slow, and it's going to take us quite a while to get these studies finished. Canadian Blood Services so far has collected over 900 units of convalescent plasma, and our sister organization has collected 1,000 units. I think, as you'll all know, the outbreak has been worse in Quebec than in most of the rest of the country. So I'd just like to say a couple of words about other passive immunity strategies. So what happens as the kind of next steps if we can show that convalescent plasma actually works? Well, what we'll probably end up doing is creating a product that makes the level of antibodies more consistent across the donations and across the product that patients are receiving. And we do that by pooling together the COVID convalescent plasma and isolating the immunoglobulin from it. This is actually done by commercial plasma fractionators, and they have gotten together to start to make this kind of product. And there were some early clinical safety studies being conducted that started in August, and should have some data results coming out of them by October or November. So this product is pooled, and as a result, it has a much more predictable profile. You have a lot number that has a certain antibody level in it. And the other important thing about this product is it could be used for prophylaxis. In other words, using it to treat healthcare workers who've been exposed to COVID-19, but have not yet come down with the illness themselves. There's also an equivalent passive immunity that's actually a model whereby you can actually grow up antibodies in a laboratory using the antibody producing cells from people who have recovered from COVID-19. So these products are still in development. There are some early safety studies, again, being done with some of the earliest products, but we don't expect to see these products realistically probably for about another year. I'd just like to say something about these antibodies that we're measuring and the use of these titers and what this all means to us as herd immunity, and the idea that our population would ultimately be vaccinated and need to make antibody responses to this virus. We have seen in all of the work that we've been doing collecting convalescent plasma that the immune response is really variable from one person to another. Most patients are making an antibody against that spike protein or the docking protein in fairly high titer, but not all of these antibodies actually neutralize or kill the virus. The intensity of the antibody response and the length of time that it lasts seems to be roughly correlated with how severe the infection was, how sick you were. However, we also see that there are patients who did have a positive COVID-19 test, but they have relatively little antibody a month after they've gotten better. So the amount of antibody you retain in your system is very variable from one person to another. It looks like these neutralizing antibodies, the virus-killing antibodies, are present in our plasma for somewhere between a few weeks and a few months. And we don't really know what that's going to mean as we start to develop vaccines, but there's certainly a lot of investigation going on and a lot of testing. So for the convalescent plasma donors, I'm sorry, I've already showed you this slide, but just to remind you again that you can, if you know people who've had COVID-19 who you think might be interested in being plasma donors, or if you have had it yourself and are interested in being a plasma donor, please go to blood.ca and register, and we will contact you. Just like to show this one last slide, because this is essentially one of our happy donors and one of our staff people who's actively working to help us with these COVID plasma, convalescent plasma collections. And these folks are making a big difference for this clinical trial. So I'd just like to say thank you very much for your attention.
Well, thanks, Dr. Devine. That was a really excellent talk on Canada's progress in developing convalescent plasma for COVID-19. And just a reminder to all the participants, please put your questions into the YouTube chat box, and we'll have them read out to the speakers. Now, I've got a question for you, Dr. Devine. Is how long does I've got two questions for you actually. So, how long does the COVID-19 convalescent plasma last in storage for, and how is it stored?
Yeah, so within 8 hours of collecting the plasma, we freeze it. So it's delivered to hospital sites frozen, and it can be stored frozen for about 12 months.
Okay, and the other question I have for you is 500 milliliters of plasma. Those of us that worked in ICUs, that's a lot of volume for somebody on a ventilator. And has there been any problems with that large volume of plasmapheresis plasma?
Yeah, so it's given quite slowly. So this 500 mils is there's attention being paid to not giving circulatory overload to sick people, and so the infusions are done at a quite slow rate. So it's over a period of at least 4 hours that the infusion is given, and so far we've not been hearing reports of circulatory overload.
Okay, very good. And so a question for the audience, do you know what percentage of patients who recover from COVID-19 actually develop neutralizing antibodies, and that could be used for convalescent plasma?
So we do have data, obviously, from the patients that have recovered that have come in to donate plasma. And we're seeing when they first appear that about 80% of them have neutralizing antibodies that are at least at a titer of 1 in 40. But then we see that over the course of about 3 or 4 weeks, most of those people start to see a decline in their antibody titer. Not everybody, but most of them. And so by 4 weeks later, after they've signed on to donate, we're seeing only about half of people have been able to maintain a titer at a level that we would use their plasma for a clinical trial.
Great. And another question is CoV SARS-2 spike chemistry so specific that COVID convalescent plasma antibodies do not interfere with good endogenous and exogenous, for example, medication entities that bind to ACE2 receptors?
So that's a good question. I think we don't really know that yet. We have not, the trials that are operating in Canada do not have an exclusion criterion for patients being on other medications. So we hopefully will be able to sort that out when we get the full 1,200 patients into the study. But at the moment, we don't know the answer to that.
Great. And a question from Michelle Schulzberg. Could you please describe the distinction between IVIG and convalescent plasma?
Sure. So IVIG is antibody derived from the plasma of anybody who wants to go donate plasma. So this is not from people who have recovered from COVID. There is a clinical, well, I think there are 2 clinical trials operating right now that are actually trying to understand if you give intravenous immunoglobulin, can you somehow modify the host immune response to the COVID virus? Those trials are still ongoing, so we don't have the answer to that yet. But it's a very different concept from the idea of giving convalescent plasma, where the antibodies in there are directed against the virus.
Okay, very good. And another question from the audience: does blood type of donors impact the number of neutralizing antibodies?
Oh, this is a good question. So there's some interesting literature that's still lacking enough data to really understand the impact, that would suggest that your blood type may influence whether or not you get COVID and whether or not your COVID is severe. And that's related to the fact that people who are blood group O make antibodies against the A blood group. It's just that you're responding to the being stimulated by your gut microbiota primarily. And there is a sugar on the virus that looks like blood group A. So there are kind of epidemiologic evidence in the literature that people who are blood group O are disproportionately underrepresented in populations of people who come down with COVID disease, and that when they are there, their disease is less severe. So there's a lot of interest in trying to tease this apart right now, but the studies really aren't that great. And so there's all kinds of confounders in the data sets and that sort of thing. So we don't really know yet.
Okay, very good. And so one last question from you is just in your opinion, why do you think the FDA gave IND status to this product, so investigational new drug to the status so quickly to this product?
Oh, I think it's to be honest, it's a poisonous mix of science and politics going on in the below us. And I think that what you're seeing is the FDA under an incredible amount of pressure to just come up and do something. So they created a situation in which they, unlike Health Canada, which I personally think did the right thing and said, if you want to use this, you need to do it in the context of a clinical trial so that we can understand whether this is a safe thing for Canadians. The FDA allowed an open investigational new drug authorization. So all you had to do was go online and fill out a very simple form and you could get product for anybody. So they've had tens of thousands of patients treated with what are called emergency INDs on a one-off basis in the United States. And there aren't really any data that can inform whether or not convalescent plasma worked. So just a very different approach. And I think that we did the kind of calmer, more sensible thing on this side of the border. Fortunately, our politicians seem to stay away from the science, which is good.
Yeah, very good point. So another question from the audience is, the antibody titers decline, but can immune cell memories work to produce antibodies again in a second exposure to COVID-19?
Yeah, I think the answer to that is generally yes, although we do now have a couple of examples of people who have caught COVID a second time. And so clearly their cellular immunity isn't working well. So it does seem to be possible to get it again. But that's a very rare set of circumstances. So generally what we're anticipating is that even if your antibody level is falling, like it would with most things that our bodies make antibodies to, eventually the titer drops, but you have your cell-mediated immunity that will rev up your antibody again if you get re-exposed.
Right. Thanks, Dr. Devine. So
my pleasure.
Any more questions from the floor? If not, we'll move on to our panel discussion. But again, I'd like to remind everybody in the audience, please put your questions into the YouTube chat box, and we can have the panel also take a look at your questions and review them. So it's and again, we've had a great number of speakers here today, and just really on the leading edge of what's going on in Canada for COVID-19 research. So first of all, we'll start the panel again, and so we have Dr. Devine on, we have Dr. Scholzberg, Dr. Tien, and Dr. Langley, and I'd like to open the floor with just a general question to the whole group is when do you think Canada is going to have a handle on COVID-19? When can we expect to have good therapies and a good vaccine that's going to make a difference, and so we can continue both normal operations in the military, at Orange, and in healthcare, as well as our lives in general?
Maybe I'll kick off then. So I mean, I think a large part of that in terms of our normal lives is a public health question in terms of when there's been a lull in how we've been practicing our social distancing and large gatherings. And I think when we look at on the news from our public health officials, there needs to be a push to sort of regain that initiative in terms of refraining from public gatherings and parties of such. So I think, the therapies and the vaccines are important, but ultimately this is a public health emergency, and control that'll be the primary response, I think.
I think, you know, as well as the vaccine, whatever vaccine becomes the most successful, we're going to be distributing. And, you know, I'd like to get, you know, Homer's and Joanne's opinion is, you know, how are we going to roll that out, that vaccine out to, you know, remote communities and Indigenous communities in our country? How are we going to get all those populations vaccinated?
Mm-hmm. I could go ahead, Homer.
That's a very challenging question. I mean, I think the good news is that healthcare in Indigenous communities is primarily on reserves is a federal responsibility, and Health Canada is first and foremost primary care and preventative care. So I think they have a very robust system. I think some of the challenges will be about procurement and transport, as Joanne pointed out, that the cold chain is quite extensive. And so if you need -50 for the vaccines and trying to develop a system to get these vaccines to the communities and preserving that cold chain will be a challenge.
Hey, thanks, Homer.
Yeah, so I would agree with that. And just to echo the very important point about public health measures and mitigation, I'm very fortunate to live in a part of the world, Atlantic Canada, where really we're one of the safest places in the world to be because of the public health measures that are in place and just the general population's adherence to these non-pharmaceutical interventions that interrupt disease transmission. So with regard to the rollout, there is a huge and comprehensive effort underway within the Public Health Agency of Canada and Health Canada, along with the provinces and territories, to design an overall system for this rollout. So you can think about the technical components, which we've mentioned, you know, how do you actually move the vaccine from wherever it's delivered in Canada to the ultimate place where an immunization provider is going to give it to a patient or a client. So that is a huge aspect. We also don't just need the vaccine, we need the syringes, we need the medications to treat adverse events, we need a safety surveillance system as we roll this out to identify safety signals. Another key component that we'll need is education for healthcare providers. So the ramp up to understanding a number of different vaccines, if there are indeed a different number of vaccines, will be an important consideration. We want healthcare providers to be confident in their knowledge about the vaccine that they are providing informed consent to a participant about. We want them to understand the potential benefit of the vaccine and the safety of the vaccine. If there's more than one vaccine, that's a little bit of a different challenge than we would normally have, for example, for influenza. And so we know that someone's decision to get a vaccine is very much influenced by their healthcare provider. It's almost the most important variable. Do they trust that healthcare provider? And can that healthcare provider answer their questions? So designing education rollout for all the components of the folks that will be interacting with citizens and being able to explain to them these vaccines, I think will be really important.
Okay, thanks, Dr. Langley. So a question from the audience, another question for you, Dr. Langley. What have been the most important side effects in the current clinical trials of the COVID vaccine?
Well, we don't really have much in the way of results from the phase 3 trials. The early trials look at what are called solicited adverse events in the days following immunization. And those are like pain at the injection site. That is routinely expected after most vaccines. That usually is transient and it goes away. There is an incidence of fever that's probably a little more present in the vectored vaccines, the adenovirus vectored vaccines. So your body's responding to the vector, the adenovirus, as well as to the spike protein which you're being presented with. So far, those are the ones we know about, and those are in small numbers. So we really need those phase 3 trials to be able to compare placebo to vaccine and say with confidence what adverse events above baseline we would expect.
Thanks, Dr. Langley. Question for Michelle and Dana. So transfusion can be a procoagulant. What do you think if somebody was randomized to the Concord trial and the Rapid trial, what would be the outcome? So the interaction of convalescent plasma and heparin, what would be the outcome?
It's a really good question. So the heparins are an anticoagulant because they potentiate the action of a natural blood thinner that exists within us all called antithrombin. And so there's the possibility that patients who are receiving both the combination of plasma plus heparin, that their bleeding risk might be a bit higher because you're giving in the plasma some of the antithrombin, although I defer entirely to Dr. Devine because I'm not aware of the quantity of antithrombin that exists in the convalescent plasma whatsoever. But that's one possibility. So for example, for patients who have heparin resistance, where the heparin just isn't working when they're getting, massive cardiac surgery, we sometimes give them plasma and sometimes antithrombin concentrate to overcome the heparin resistance. So that's why I'm saying that, but I'd love to hear Dr. Devine's thoughts.
Yeah, well, we don't know, right? Because we haven't looked at what the antithrombin levels are in convalescent plasma. We've not had, there's not really any reason to think it would be different from distribution in the general population, but we don't actually have data to support that.
Okay, thank you. This and a couple other questions about vaccines. And historically, vaccines are rolled out by requiring mandatory injections for military members and healthcare providers first. Do you anticipate this rollout, the rollout to take this approach? And following on to that is, what are the ethical implications at play when determining who will get the vaccine, assuming initial scarcity in the vaccine supply?
Mm-hmm. So I could answer the second question first. In terms of the military rollout, I wouldn't know. I think probably you folks would be better able to answer that. So in terms of the prioritization, so even if we had all the vaccine for 37 million Canadians in early 2021, we know that our ability to roll that program out would not allow it to occur in a week. So with H1N1, that vaccine program took about 4 to 6 months to deliver. So I'm sure there will be a ramp up in our ability to deliver vaccine. We will train healthcare providers to provide vaccine that maybe didn't do it before to increase our ability to distribute it more quickly. So the prioritization will be recommended by the National Advisory Committee on Immunization, and that guidance will be coming out in October, I understand, and they will look at who is highest risk and may look at it, for example, by occupational risk and then by potentially burden of disease, who's most likely to have this highest incidence of severe outcomes or death, and so should benefit from. So those are as partially scientific questions and partially ethical and socioeconomic questions as well. Just with regard to the whole idea of mandatory vaccination, when you use that term mandatory, it can be a very laden term that makes people defensive and upset because it implies that you're almost holding down people to give them a vaccine. And I'm not really aware of any situation where we do that. The situations in public health that are sometimes called mandatory are not in fact forcing people to get vaccine. There's situations where we require a documentation of the vaccines that they have had. So that enables the healthcare provider to make decisions about counseling those people or managing outbreaks, but it's not actually forcing people to get vaccines. And as I understand it, that doesn't happen in the military either. people have the ability to consent or not consent. And so I will, at that point, pass it to you folks to answer what will happen in the Canadian Armed Forces.
Well, I can say is the vaccinations you have the you definitely you're able to refuse a vaccine and if you don't consent to it. So, and that's just recorded in your documentation. It may have some operational effects that you can't deploy to a certain area, but there's no thing as a mandatory vaccine you can not give consent and it won't be given. Question for Michelle: is the microvascular endothelial glycocalyx can be severely damaged by COVID-19? This suggests that platelet inhibitors may be useful therapy.
Yeah, great question. So you're absolutely right that an endotheliopathy, severe endotheliopathy that likely really has its seat in the lungs is one of the things that seems characteristic of severe COVID-19. And so whether or not agents that affect platelet function, interfering with platelet adhesion and aggregation, or agents that can be directed against von Willebrand factor also interfering with platelet adhesion are being looked at. And in fact, there's an NIH clinical trial platform that is also looking at anticoagulants, starting with the comparison that we're doing as well. And then subsequent to that, the plan is to move forward once they identify which arm demonstrates superiority, and then to add an antiplatelet agent to that arm. So it could help. We don't know, but it could be.
Okay. Thanks, Michelle. Another question from the audience is: critically ill patients have a robust serological response to COVID-19, and the question is: would convalescent plasma in acute illness could it be redundant, and could it potentially risk exacerbating the innate immune system?
Yeah, I'd be happy to answer that. So the best evidence to support that is true is there was a Dutch study that was a randomized convalescent plasma trial that was started in the Netherlands in severely ill patients, and it was stopped early. And it was stopped early by the safety board because they had data showing that the antibody levels in the patients were actually higher than what was in the convalescent plasma they were giving. And so they said, what are you people doing here? You're not actually, adding any benefit. And particularly when you consider that if you give a plasma transfusion, it's going to get diluted 8 or 10 times in its volume against your blood volume. So you really are not doing anything positive here. And so that was probably the best evidence that if you're critically ill, which the study were, you're really, the convalescent plasma probably you're too late in giving it.
Yeah, and so I guess it's convalescent plasma. It's the timing of the intervention. I think it's sort of in your mind when would be the perfect time to give convalescent plasma in the ideal patient, COVID-19 patient. Where would it be in the pre-hospital environment or the emergency room? Where would be the ideal?
I think that what the literature, the studies that are publishing now are telling us is that the sooner you get that convalescent plasma into people who are symptomatic, the better off you are. And so that's really dependent upon when the patient chooses to arrive at the hospital with their breathing difficulties. But you need to get that plasma in there very shortly after admission.
And another question from the floor. This is maybe I'll put this to Homer. So not consenting to a vaccine that you're being told is mandatory by your chain of command can have a huge career ramifications, including being put on a medical category. What's your take on that, Homer?
That's a hard question. I mean, there's a significant administrative component to that question that it's hard for me to answer. I do think that from an operational point of view, if you don't consent to, if you're going to a high risk area that the incidence or the risk of exposure to whatever viral illness is significant or is deemed to be significant. It's sort of the responsibility of the chain of command to say that from an occupational health and safety point of view, that if you don't get the vaccine, you're at undue risk and that you can't deploy. So there may or may not be administrative consequences to that, but I do think from a chain of command point of view, it's important consideration that the chain of command is responsible for the health and safety of the military members under their command. And I think they're just looking out after that person for that purpose.
Yeah, and I would agree with that. We're trying to protect the member from, there's potentially lethal diseases on deployments that many of the people in the audience have been on, from yellow fever to the multitude of other viruses, hepatitis, etc. And this is due to protect the health of the force and the individual member. One other question to Joanne is, could you, we're talking about the COVID-19 vaccine with the sort of idea that everybody's going to get it and everybody's going to accept it. And can you just maybe tell what's the rate of people getting the annual flu vaccine in Canada, and how high do we have to get the vaccination rate up this year to actually make a difference?
Thank you. So, influenza vaccine is provided by the provinces and territories for different populations across Canada. So for example, in Nova Scotia, where I live, it's available to the entire population, free in pharmacies, from your family doctor, whatever. But some provinces only provide it to high risk. So that's just background to say that you can't use the entire population as a denominator. Even in healthcare workers, where we hover really across the country at about 50 to 60% uptake for influenza vaccine, that's probably not enough to provide population protection, although it could have some measurable benefit, obviously. So in the general population, the uptake varies according to age group and risk category. So for example, in people over 65, it would be a little higher than it would be in healthy people 18 to 50. And in children, in toddlers, for example, it's highly recommended for 6 to 23-month-olds, but probably only 34% of children in that age group are getting it. So the uptake is not ideal, for sure.
Okay, and we're just wrapping up our panel discussion. We have 3 minutes left, and I'd just like to get everybody's thoughts on, what's the silver lining in this COVID-19 pandemic, if there's any, in terms of research and knowledge about the immune system or transport or hemostasis. What's in everybody's mind? What's going to be the silver lining in this in your individual area of study? And I'll start with Dana.
Yeah, that's a good question. I think that this has driven us to really think through what maintenance of the blood supply means in the midst of a pandemic, and recognizing that this probably isn't the last pandemic. And so our practices in clinics, our need to maintain physical distancing, the idea that we seed in the donating public, that donating blood is still an essential thing to be doing in a pandemic so that we can maintain blood supplies. We've in my view, dodged a bullet, that we've got a pandemic of a virus for which blood products are not needed to treat people who have it. But, God forbid they were, we would be in some trouble. And so, I think we've really learned had the opportunity to think about some of these things in advance of finding ourselves in that situation. So to me, that's the silver lining.
How about you, Michelle? What's the silver lining for you in this?
Yeah, I'd like to echo a lot of what Dr. Devine said as well. I think there's been tremendous camaraderie and scientific collaboration. People coming forward and also tremendous collaboration from various hospitals across the country and internationally. So as part of the RAPID trial, the majority of the hospitals that stepped forward indicated that they would like to use their own internal funding to support the clinical trial. And that, I think, just speaks to really just unbelievable generosity of spirit and people's willingness to help patients. So that has been a huge silver lining for me. Obviously, this is a very distressing time for everyone, but it's also an incredibly inspiring time. And I think that Dr. Devine also emphasized that we should probably have a lot of Canadian pride because we're all operating, I think, in as constructive as possible way as we can right now, which is really important. So that's been the silver lining for me. I think from a scientific perspective, from a clinical perspective, the silver lining in my involvement at St. Michael's Hospital has really, transformed my perception of what we're capable of. And I know, Dr. Beckett, you're aware of this too. We're changing policies on a dime. We're restructuring the entire hospital. We're moving quickly, and it shows us that we're tremendously flexible and nimble. And that's also really good to know.
Thanks, Michelle. Homer, how about you? What's the silver lining in this for yourself and Orange?
I think it's a reminder about how important frontline staff are to the healthcare system. The military ethos, I think people recognize the appreciation for frontline troops. And I think what we're seeing during the pandemic is the appreciation for self-sacrifice and the willingness to sort of run towards danger for frontline healthcare professionals, both pre-hospital and in the hospital. I think it's a reminder to leadership that we have to have the backs of the people at the front lines.
Thanks, Homer. How about you, Joanne? What's the silver lining from a vaccinology point of view?
Two things. One is the tremendous impact of non-pharmaceutical interventions on reduction in disease transmission. So all these things we do, the personal protective equipment, the distancing, the way we move within buildings, the air supply, we're going to be learning so much more that will be applicable to all infectious diseases, I think. And then the second thing is echoes what's previously been said, is that in the case of vaccine production, the acceleration in development of every aspect of how you bring a vaccine from concept to having products you can roll out in mass programs has just been tremendously increased. And the way that we do that together, and just the power of many minds working on the same problem and how that can advance science is really humbling, I think.
Oh, great. Well, thanks, Joanne. And I'd like to thank all of our speakers and for coming and spending their time with us today and recording their videos just before this. And I think it gave us a really good picture of the Canadian contribution to the fight against COVID-19. And I'd also like to thank the audience for coming out today and taking time to listen to Simvar's presentation, and especially to those who submitted questions. Some of the questions were really excellent. And I think this is a reminder that to all of us that how important Simvar is linking up people from the military and Veterans Affairs and from those doing defense veterans research with civilian collaborators. And I think in this time where it's hard to get together, I think this just shows again how important Simvar is moving these milestones further. So with that, I would like to give the floor back to Dr. David Pedler, the Scientific Director for Simvar, and I'd like to allow him to make his closing remarks.
Thank you for joining us to kick off the virtual symposium series in such a spectacular way. I want to extend a special thank you to Dr. Andrew Beckett, Colonel Retired Homer Tien, Dr. Joanne Langley, Dr. Michelle Scholzberg, and Dr. Dana Devine. We have all learned so much from you today. I would also like to take a moment to thank Calian for sponsoring today's event. We are very grateful for your continued support of Simvar and for your commitment to helping us serve those who serve us. Before we go, I just want to add that we are thankful to our presenters for sharing their expertise on a topic so relevant to all of us right now. To all of you and your families, please stay safe and be well. Thank you.